So let's start with the material in this module on antibacterial agents. The whole research on antibacterial agents actually starts from the concept and understanding back in the 19th century that microorganisms were often associated with disease. And that started a whole effort to use agents as a disinfectants and germicides. And so we were looking at things that could be used to um, disinfect surfaces, and especially in the um, hospital settings or doctor's offices. And those included dyes, phenols, ethanol, other surfactants. Um, and really built on the idea that, unselectively killing microorganisms. The revolution then happened when doctors and scientists started to looking for agents that could be used systemically. The first so-called chemotherapeutic agent, where a chemical is used in the body to cure a disease, was salvarsan, which is against syphilis, which is a transmissible disease caused by a bacterium. And as we'll see, the whole concept of this kind of compound starts from the idea that we can actually stain the bacteria, or rather attach something covalently to their surface, and that in turn may result in their in killing them. So the first antibacterial agents were actually what we call chemotherapeutic agents. So they were compounds, chemical compounds that can prevent the growth of microorganisms, or that can kill them inside the host. And very quickly, we started then from chemotherapeutic agents to use antibiotics, which are natural compounds, and then they can be altered by either semi-synthesis or complete synthesis. And we'll get back to that, but the whole field of antibiotics was started by the discovery that penicillium colonies grown on Staphylococcus petri dishes were undergoing lysis in the area around the mold. So what are the characteristics of a chemotherapeutic agent that we may want to use for treat infective diseases? So it needs to be, usable inside the body, so not toxic for the host. It needs to be efficient at small concentration and need to have selective toxicity. A selective toxicity in terms of, well, not harming the host, but also in terms of which microorganisms it targets. And so we'll distinguish between agents that are broad spectrum, so they're active against a wide range of infective agents, and agents that are narrow spectrum, so they have a very defined specificity. First of all, let's start also from reminding us of what a bacterium looks like. In general, in terms, bacteria are single-cell microorganisms, and they are prokaryotes. So that means they don't have a membrane membrane that surrounds the genetic material, so they don't have nucleus. They don't have mitochondria, which are small and microorganelles, confined, surrounded by a membrane. And in fact, the current theories say that mitochondria are actually a product of symbiosis between prokaryotes and eukaryotes. They don't have any membrane-bound organelles, so they only have cytoplasm, which contains the nucleic acid, which is a circular DNA chromosome, a single if you wish, that is in the center. Genetic material is also stored in plasmids, which are circular bits of DNA encoding for particular properties or proteins. And then it contains cytoplasm. Um, and in this case, this is E. coli. We can also identify other agents. So we have a plasma membrane, so the membrane that surrounds the cell. We have a cell wall, And then we have a capsule, which is further surrounding the cell wall. E. coli is one of the most widespread bacteria. It's a so-called commensal bacteria, so bacterium. So it's found in the GI tract of anything that is a vertebrate and warm-blooded, so including humans. It generally is harmless and actually is implicated in the synthesis of vitamin K2 in the GI tract. It can be dangerous though if it passes, the GI tract for some reason, and especially if you know basically outbreaks of E. coli infections are found when food is contaminated by fecal um, traces, and that happens especially with vegetables that have been cultivated close to a farm environment. Some strains are virulent, and the virulence actually, is expressed in a plasmid that can be exchanged very quickly between E. coli strains. And in fact, some E. coli variants can express a toxin similar to the Shiga toxin that causes important GI tract problems. Um, so E. coli is a gram-negative, and we know that because that means it has a small cell wall. Um, it's about 2 micrometers long and about 0.5 micrometer in diameter, and it possesses a flagellum and it uses that to move around the environment. So because it can pick up plasmids and therefore genetic traits very easily, E. coli has also been a workhorse in expression of recombinant proteins and so in molecular biology. So besides the plasma membranes, then we have a cell wall, and then we have this capsule that contains an outer membrane. And as we'll see, basically, these features make gram-negative resistant to certain antibiotics. So let's talk more generically about how we classify um, the antibiotics and antibacterial agents. And so we can cover them and classify them according to their molecular target and site of action. So we'll see things that act on the cell wall, on the cell membrane, on DNA, on protein synthesis, and on metabolic reactions. And here's a more in-depth view of what we just saw in the previous slide. So we have the cell wall synthesis, which is inhibited by a bunch of antibiotics, including penicillins and um, cephalosporins. DNA transcription into RNA is inhibited by quinolones and rifampin, as we'll see. And then translation for messenger RNA to protein is inhibited by chloramphenicol, erythromycins, tetracycline, and streptomycin, which generally act by binding to the ribosomes. We also have other kind of more blunt injury to plasma membrane, and that happens with polymyxins, which are very generally effective antibiotics. And then we have other agents that selectively inhibit enzymes that are essential for bacteria but not for humans, and those will be the sulfa drugs and trimethoprim, for example. So the most important classification and distinction that we do with bacteria, and that helps us also in evaluating whether a bacteria are susceptible to certain agents or not, is the so-called gram staining. So this was invented by Hans Christian Gram in 1884, and of course he had little idea what actually was happening, but is still used to this day. And so what we see here is gram positive and gram negative bacteria in the same culture. So how does the staining work? It's actually pretty simple. It works in several phases. So first, bacteria are stained with a dye called crystal violet, which is then oxidized with iodine, and that stains then purple. Then there is a destaining step with alcohol, which basically serves to remove all the unbound dye. And so the gram-positive bacteria will retain the stain because they contain a thick peptidoglycan wall, which reacts with the dye and retains the dye. Whereas a gram-negative bacteria, which has a much smaller cell wall and also surrounded by a membrane, do not do that. And then to help visualize in the contrast and increase in contrast, we often use a secondary dye, typically safranin, which stains gram-negative bacteria by a different mechanism, so they react with lipids, and helps us visualizing the difference between positive and negative. In this slide, we are observing the difference between gram-positive and gram-negative bacteria. So gram-positive, which we saw stain positive with gram-positive with the gram stain, contain the membrane of the plasma membrane here, and then contain a thick peptoglycan layer. And we see how the composition of this peptoglycan layer is carried out and how the synthesis is carried out. Gram-negatives, on the other hand, have a plasma membrane that contain a very thin peptoglycan layer. There is then a periplasmic space here, and then there is an outer membrane, which is again a um, bilayer, phospholipid bilayer, which does not stain with the gram reagent.